Today we'll be covering two topics. First, we'll talk about gene expression. Then we'll take a look at DNA mutations. First of all, genes are expressed when they are displayed in physical traits. It turns out that cells respond to their environment by producing different types and amounts of proteins. For example, have you ever seen a green potato chip? It's like, oh, gross, don't eat that one, it'll hurt you. No, there's nothing wrong with the potato chip. What happened was the potato stuck out of the ground a little bit, and the part of the potato that stuck out of the ground was exposed to sunlight. The environment, the sunlight, caused the potato to begin producing chloroplasts, which then made chlorophyll, which then caused it to be green. And so that's why if you find a green potato chip, it's actually not a problem at all. It just got exposed to sunlight. So you'll see that the environment caused the cell to respond differently. So how did the root produce chlorophyll when it's the leaves that are supposed to be having that? Well, remember, every cell in that potato plant, except perhaps egg and sperm cells, has the exact same DNA. But they're different based on which genes are expressed, which ones are used in the plant. So in the leaves, they'll use the genes for the chlorophyll and chloroplasts. In the root, they'll use the genes for storing carbohydrates for energy. Every cell has all the information to make the entire potato plant. It just uses different pieces of it. Then, when the cell produces that particular protein, it leads to a particular function. For example, chlorophyll is used to make energy from sunlight, or a particular phenotype. Chlorophyll will cause it to be green. The proteins that are made can be structural, part of the cell materials, how it's built, or they can be functional, such as hormones carry chemical messages, enzymes help reactions happen faster, and other chemicals involved in cell chemistry will cause the cell to do different things. There's a cause and effect relationship between environmental factors and expression of a particular genetic trait. It's not just in potatoes that it happens, it's in everything. For example, you may or may not inherit a gene that means lung cancer runs in your family. However, if you chew tobacco, you are more likely to express that gene and develop lung or mouth cancer. Once again, skin cancer may run in your family, but if you stay in the sun and get burned over and over, you are more likely to express that gene for skin cancer. On the other hand, there are other things in your environment that can help prevent that. See, you need some sun so that your body can make vitamin D. But too much sun to where you burn yourself can cause skin cancer. So how do you prevent this? Well, taking folic acid or eating foods with folic acid will help your body make new cells and fight against skin cancer. By the way, folic acid is a B vitamin. Here's another example. Diabetes may or may not run in your family. However, if you eat a very sugary, processed diet and you exercise very little, you may develop diabetes. In fact, you're going to be more likely to develop diabetes than you would otherwise be. However, if you eat a healthy diet with lots of fresh fruits and vegetables, and you exercise in a healthy way, then you are much less likely to get diabetes. Phenylketonuria, or PKU, is a genetic disorder in which people do not digest proteins correctly. They build up in the brain and cause early death. People who have PKU must avoid protein as much as possible. The more protein they eat, the more it hurts their body and the sooner they die. The less protein they eat, the longer they live. However, you have to have protein to build muscle, make your immune system, make certain hormones. So it's not a good thing to have. And you already know that heart disease is very strongly affected by whether you have a healthy diet and exercise or a poor diet and you don't exercise. So all of these are environmental conditions that can affect the expression of the genes that you may or may not have. And one random fact, the same 5 nucleotides, A, C, G, T, and U for RNA, are used in the DNA and RNA of all organisms, every living thing on Earth. Plants, animals, bacteria, even viruses have either DNA or RNA, and most people don't even consider them to be living. Now let's take a look at some DNA mutations. Mutations are changes in DNA coding. They can be deletions, additions, or substitutions. Mutations can happen randomly and spontaneously, in other words, all on their own, or they can be caused by radiation, like nuclear radiation, or by solar radiation, which is not nuclear, but it means light. So ultraviolet light is a type of solar radiation because the light radiates or shines out of the sun. And mutations can also be caused by chemical exposure. If you're exposed to certain toxic chemicals, those can cause mutations. Mutations work by changing the amino acid sequence. See, if one of your 3 letters is different, that can give you a different amino acid. Then, putting in a different amino acid changes the protein. The protein may or may not function the way it did in the past, especially if it's a significant change. Then, having the protein be off changes the phenotype, the physical appearance. So something won't look or act right. Now, most mutations cannot be inherited. Only mutations in your sex cells or your gametes, egg and sperm, can be inherited. Mutations in your skin cells will not be inherited because you don't pass on your skin to your children, you pass on an egg or a sperm. Let's look at some different types of mutations. First of all, you can have a point mutation. One little point in the DNA is mutated. One base is exchanged for another. So either A, T, G, C, or U is switched with one of the other letters. Here are some examples of point mutations. Substitution is when one letter is substituted for another. Now, let's pretend that instead of A, G, T, C, and U, that instead we have the entire alphabet to use when making amino acids. So let's say that you had a piece of DNA that said the big fat cat ate the wet rat. Okay, well, if you change one letter, so G was changed to Z, that's a substitution. And now the sentence says the biz fat cat ate the wet rat. So this word has turned into nonsense. The rest of the sentence still makes sense, but what's a biz cat? Who knows? So that can change the sentence. In the same way, when you have a piece of DNA and one of the letters, let's say this T changes into a C, well, now this second amino acid will be a different amino acid than the one that originally was included in the protein. Another example of a point mutation is an insertion. An extra letter is inserted into the sentence. For example, if instead of replacing the G with a Z, we just add the Z, that shoves all the other letters over by 1. Which all of a sudden gives you nonsense. I mean, this doesn't even make sense anymore. This is called a frame shift mutation. You can see the frame just shifted over completely, and it no longer groups the same 3 letters together. Here's an example of an insertion using DNA. Between the C and the T, an extra C has been added, shoving these over. So instead of TAA, CTG, CAG, and then you have a leftover G and T. It now says TAA, okay, that amino acid is the same. CCT, nope, that's different. GCA, different. GGT, different. Wow, lots of things just changed. A third type of mutation is a deletion. This is another point mutation because one letter is deleted. Let's say we deleted the letter E from the word the. So now the entire sentence shifts left, another frame shift mutation, and once again you get a lot of nonsense words. Now those are all point mutations, but there are also other types of mutations. One is a repeat expansion. It takes one of the three-letter codons and repeats it and expands the sentence. This is what happens in Huntington's disease. Huntington's disease usually shows up in a person's 30s or 40s, causes shaking, and causes mental decline. This is caused by a repeat of one codon over and over at the end of a chromosome. The more often that codon repeats, the more severe Huntington's disease is. Another mutation is an inversion. It inverts or flips part of it backwards. So if we take the word cat 8 and wrote it backwards, cat 8, then that reverses these two groups of letters and switches them around. So that messes up these two amino acids. Here's an example of an inversion. You'll notice they took the letters T, G, C, A and wrote them backwards, T, G, C, A, which of course changes two of the amino acids. You can also have a large-scale deletion. For example, in this sentence, 1, 2, 3, 4, 5 words have been deleted, greatly shortening this sentence and completely changing the meaning. In the same way, you can either have a single base that is deleted or a whole group of bases that are deleted. Furthermore, an entire half a gene or a gene can be deleted, an entire section of a chromosome, or even an entire chromosome can be missing. When an entire chromosome is missing, this is usually caused by non-disjunction. Remember, during meiosis, the pairs of chromosomes are supposed to be separated or disjoined. When they don't separate, non-disjunction, then you end up with one egg with an extra chromosome and one egg with not enough chromosomes, causing problems either way. If the egg has extra chromosome 21, you have Down syndrome. If it's extra chromosome 13, it's cri du chat syndrome, or cry of the cat cry syndrome. Finally, just like these point mutations can change a single letter, they can also change a group of letters and be a larger mutation. For example, instead of substituting one letter, you can substitute a large portion, and instead of inserting one letter, several letters can be inserted, and instead of deleting one letter, several letters can be deleted. Here's an example where an entire 3 letters are substituted instead of just one letter. And as we have said, there are many other large-scale mutations that can occur. One is called a copy number variation, or CNV. This is a mutation where large chunks of DNA are inserted, repeated, or lost. These regions of DNA can be between 10,000 and 5 million bases long. That's a lot of mutations. Genes can be duplicated. You can have multiple copies of a gene. Large regions of the chromosome can be deleted. If you had two copies of a gene, one from your mom and one from your dad, you could lose one of those copies. You could lose both copies of that same gene. A part of DNA could be picked up and moved to a different part of the string of DNA. And an entire extra chromosome can occur or can be missing. So as you see, there's a lot of different ways for mutations to occur. We're probably pretty fortunate that we don't have any more mutations than we already do, because mutations almost always are bad. Very few mutations are beneficial. The only one I know of is sickle cell anemia. And when I say beneficial, 2 copies is horrible. 1 copy of the mutated gene that causes sickle cell anemia will help prevent malaria. However, 2 copies, and now you have sickle cell anemia, which causes all sorts of problems. So mutations in general are very bad things, and it is very, very rare for a mutation to be even partially useful, like sickle cell anemia.